Alright, so Dr. Orlando here. We're going to do another vidal on DNA, and this is DNA translation. In previous vidal we did DNA transcription. So DNA translation is when we use RNA we use it like a recipe to make a protein. It's a recipe for a protein. All of this occurs in the cytoplasm. Okay. And we call it translation because we go from a code made up of nucleotides in RNA to a protein made up of amino acids. So we're translating. We're going to a different language altogether. All right, so let's look at how this happens. Okay, so I'm going to use the picture from your book because it is just too difficult for me to go ahead and try to draw. So I want to label a few things here and make sure that we understand where we're at in the cell. So to begin with, here's the picture of the cell, and notice that we are in the cytoplasm, and this is a ribosome here, shown in green. Okay, and the ribosome is where the mRNA strand is going. So let's label this here. This is our mRNA. And then these kind of greenish color blobs, these are our ribosome. And a ribosome is made up of two parts. There's a small subunit and a large subunit. And initially they're near each other, but they're not necessarily attached to the mRNA. We also have, to make sure we know what things are here, let's see if we have any. These structures here that are here, here, and here, and then you can see other ones at the bottom here. These are, let's do undo. Let's do this one. These are tRNA. So when you look at them, that coiled up yellow business right there, that's the oh my goodness. tRNA. And then this is the amino acid that they're carrying. tRNAs are a sequence of nucleotides, right? Because they are RNA, and RNA is a sequence of nucleotides. Well, there are three most important nucleotides that are part of their sequence, and they are known as the anticodon. So looking at tRNA, this is called the anticodon. Okay, and it's these three bases right here, here, and here. Okay, those three nucleotides in a row. And we'll see why they are so important. But for now, I need you to believe me, accept that the anticodon is correlated to the amino acid. So certain tRNAs will only carry certain amino acids, and it's because of this anticodon that it becomes specific. Okay, so these are our players right now. We've got the ribosome, small unit, subunit, large subunit of the ribosome. We've got the tRNA carrying the amino acids, and we've got the mRNA. All right. I just wanted to clean up my picture here so that we can go ahead and draw on it. All right, so I went ahead and drew in a code. This is the same one that's in your textbook. It's nice to use theirs because we know it's going to work out right. So you can see that the mRNA has that sequence there, and it has this area here that's green, and notice there's no amino acids attaching to that. We call that the leader sequence. And you can think of it like a shipping label. It leads the mRNA to the ribosome. It's not going to end up being part of the actual protein, though. The first amino acid to be brought to the chain will be placed right after the leader sequence. So the first thing that's going to happen is this step called initiation. We're going to initiate translation. So the first 3 bases that will be read will be complemented by a tRNA molecule. Okay. How do they do that? How do they know where to go? Well, this first 3 bases, and as we know, as we said in class, we should mark off our bases. Helps us keep them separate. We call these 3 base sequences codons. Okay. And this codon, AUG, if you look up in your chart, and we'll get a chart in just a minute, you'll see that it codes for a methionine, a special kind of methionine if it happens to be the first one. And the taxi, the tRNA, the transfer RNA, will bring methionine there. And it'll bring it there because of that AUG. And the anticodon complements it. So if it complements it, it has affinity for it. So it has affinity for it, therefore it's attracted to that spot on mRNA, and those bases will pair up happily. So there they are, paired up. So we've got the mRNA attached to the small subunit. We've got the first tRNA bringing in the first amino acid, methionine. And then the large subunit will attach, almost like clamping into place. So then we can move to the next step. The next step is called elongation. We're going to elongate the protein. Okay, so here we go. We're going to bring in the next tRNA. So I'm going to write in the code again on this one. All right, got everybody labeled now, so we can move along and elongate our growing protein chain. Right now, we just have 1 amino acid on board, so we're going to add the next one. All right, so we're going to look at the codon. Remember the codon. Codons are on the mRNA. They are those 3 base sequence. So here I've got 3 codons written out for you. Okay. So this codon, GGA, is the next one that is going to expect an amino acid be brought in. Okay, so GGA, what would the anticodon be here? The anticodon would complement GGA, so it would be CC. Remember, A can't pair with T because this is RNA, so CCU. Okay. Now, how do we know what amino acid that means? Well, if you want to find out what amino acid is, you have to look at the codon, because that's the kind of charts we have. We have codon charts. So the one in your textbook and the one in the back of your lab book are fine to use. So we will go ahead and look up and see if we can figure out what GGA codes for. All right, so how do you use this chart? We are looking for GGA, that's the codon. What amino acid would this transfer RNA be carrying? So you start here with the G's, because it says first letter. This is the first letter, so U, C, A, and G. We've got a G. And then we move over to the second letter. Use these columns to find the second letter, which is a G. So we know we're going to be in this box. We go that way to that way. Okay. Now that's pretty easy because everything in this box is going to code for glycine. But if it didn't, as you can see, sometimes there's more than one amino acid per box. If it didn't, you would just work your way down and go G, G, A, and you'd find it and figure out which one it points to. Okay. So it's going to code for glycine, and the abbreviation is GLY. All right, so let's go back and we'll write G-L-Y. Okay, so there it goes. It's going to bring in the tRNA, is going to bring in glycine. Okay, and it's going to put it right into that spot on the large subunit. And what that's going to do, this ribosome's acting like a giant enzyme, is going to make sure that those two amino acids are close enough together so that they can form a bond, a covalent bond between them. Okay, so go down to the next one here. All right. And I'm not going to draw in this is still initiation. Oh, no, excuse me, not initiation. Goodness. Elongation. Okay. So we had got the methionine and glycine, as we just figured out, go in here. I'm going to go ahead and add in our codons and anticodons at this point too, just to make it all pretty picture. All right, so I put in the bases in the mRNA strand. I put in our anticodons on our tRNA. And now I want to point out the big important thing here in this picture, which is, notice that the methionine and the glycine formed a bond. So we've got we need a darker color than that. We've got an amino acid bonded to another amino acid, and now we've made a covalent bond, and a special kind of covalent bond that binds amino acids is known as a peptide bond. Okay, that peptide bond is covalent. And at this point, just as a kind of review, always remember that proteins made up of amino acids, if it is a short strand of amino acids, we call it a peptide. A long strand of amino acids, then we call it a protein. All right? And since this is only two, we could call it a dipeptide, dipeptide. But it's going to get longer, so we're not going to call it that at this point. Okay, so now we've got that peptide bond formed between the two amino acids. Good. All right, continuing with elongation here. The next tRNA is going to bring in the next amino acid, and when that happens, everybody's going to take a step to the side, and notice that now glycine has moved over, methionine has moved over, and now we've made room for the next guy. So let's figure out what amino acid should go here. Remember, we use the codon on mRNA to look on our chart. So the codon is UGU. And so we'll go look at our chart. Let's get rid of this stuff here. Okay, UGU, so we're going to start with the U's. Okay, so we know we're going to be in this row. Then we need G. Let's write this bottom at the bottom, where we're doing UGU equals what amino acid? Okay, so U, G, U. So let's go with, we know it's going to be in this box, and there's UGU, cysteine. Okay, CYS. So let's go back. And we know that this is going to be CYS. Okay? And notice that they made a glycine and cysteine made a bond, a peptide bond, and now they will take a step to the left, okay, in this picture. And then the next one will come on board. So let's just leave this picture here and figure out what's the next one to come on board. Again, we'll use the codon. AAG is what we're looking for. So we'll clean this up again. This time we want AAG. Okay, so AAG, the first letter is an A. Second letter is an A. So that's here. So we're looking at this box right here. And the third letter is a G. So AAG, lysine. Okay, so we go back. And we know that this is going to be lysine. Okay? And it will come on, as soon as these guys take a step to the left, lysine will come on board and make a bond with cysteine. So this process is going to keep going until termination. Termination means to stop. So how would this end? Okay, it ends because, and it is released from the ribosome because of this sequence here. So the last sequence that happens to be on our mRNA strand happens to be UAA. Okay, and all along the way we've added those amino acids. Now we get to one that's UAA, and there's no tRNA for this. How did that happen? Well, let's look at our chart and see how that happened. We're looking for UAA. Clean this up. Okay, so UAA, let's go with the yellow here. We're going to go with the U, so we're going to go here. We're going to go with A here. And if we circle UAA, we find it codes for a stop. So there's no amino acid associated with the codon UAA. If there's no amino acid associated with it, it's considered a stop. And the protein chain, the peptide will be released from the ribosome. Okay? What happens from there? Well, that protein, a lot of things could happen, as we know with proteins. That protein could go be modified in the ER. It might just get used. It might go do its job. It might get packaged at the Golgi. Maybe it's going to be shipped off. Okay. Those are some of the big things that could happen to that protein. What happens to the mRNA molecule? Do we just throw the recipe away and start over? No, we reuse this. How many times? It depends. It depends on the protein and the signals that the cell is getting. So that recipe will be used again, that mRNA will be reused on another ribosome, maybe the same ribosome, to make another protein. The exact same protein, but another one of the protein molecules. Okay, so that's it for translation. So if you have any questions, please feel free to contact me. Have a good one.